Your erythrocytes, or your red blood cells, are going to be the highest concentration of the formed elements. What you're seeing here, every one of these round red things are red blood cells. So if you notice, that's the vast majority of what you're seeing in this blood sample. These other parts, this one and this one, those are going to be white blood cells. While these other smaller pieces like this and this one, those are your platelets. So really, erythrocytes have the highest concentration of all the formed elements in your blood. Now, erythrocytes are filled with hemoglobin. It's a protein. Now, hemoglobin can be abbreviated as HB, but hemoglobin itself is a 4-chained protein meant to carry gases. It's containing several different proteins like spectrin, for example, besides hemoglobin. Now, the reason I'm mentioning the spectrin is because spectrin itself helps provide flexibility and the ability for that hemoglobin, as well as the rest of the red blood cell, to kind of change shape, to squeeze through places, get some flexibility going. The idea is your red blood cells have to go through every capillary in your body, every tiny little vessel. You don't want them to get stuck, which means if they get into a tight curve, they better be able to kind of bend to match that curve. Spectrin allows that to occur. So a lot of hemoglobin, but you also have spectrin giving the flexibility and changing of shape. So looking at these erythrocytes, you can kind of see how they're looking like almost a donut shape. Now, that's not really called a donut shape, it's called a biconcave disc. The biconcave disc means the center is flattened out. Look in cross-section. You'll notice right there, the ends are thicker, the middle is very, very thin. That's there to maximize surface area. Maximizing surface area allows for maximal transport of gases and oxygen on and off. So if we're unloading and basically absorbing oxygen, and then offloading diffusion of oxygen, or for carbon dioxide for that matter. But this also is important to note. See that one that's circled right now? That's in a capillary. That means it's taking up almost the entire width of that capillary. If all of a sudden we've got that sharp turn, you better make sure that thing can bend. That's why we have that spectrum. And down here, you'll notice a lot of little tiny red blood cells on a tip of something. Well, tip of what? That's a hypodermic needle, to show you just how tiny those red blood cells are. That's just the one little edge tip of that needle, not the whole thing, just the one tiny corner. So erythrocytes are very, very small cells here. Now, as I mentioned, biconcave shape. That was that thicker outside, thin inside, maximizing surface area, allowing them to kind of bend and be flexible. It's greater than 97% hemoglobin. You're saying, well, greater than. Well, some are 98, some could be 99. There's a little ranging there, depends on the cell. But that is not looking at what the water count is, as they could potentially get full of water sometimes. The remaining 1, 2, 3%, looking at the other proteins, like spectrin, for example. There's no mitochondria. ATP is anaerobic. All right, put that in perspective. Why is that important? Well, if you go back to cell biology, mitochondria are going to allow for ATP production using an aerobic pathway, which means they're taking oxygen in and using that to make ATP. There's no mitochondria here, because do you want to use up the oxygen that you're transporting, basically your cargo, before you get to the destination? No. So the cells use an anaerobic process to make sure all that oxygen, all that cargo, gets from point A to point B, and not using it up along the way. So here is what hemoglobin looks like. You'll notice that in the center, this is one hemoglobin molecule. The whole thing's hemoglobin. You're going to have 4 chains. Each one of these is a different chain. 2 alpha chains, 2 beta chains. Now, in the center of each chain, though, is this iron molecule. All 4 chains have an iron molecule at the center. So if you're looking at the more chemical view here, Fe is the iron. That's where the iron is, right in the middle of the entire chain. Oxygen binds to the iron. That means each hemoglobin can bind 4 oxygen molecules at maximum. That's saturation. Now, will every blood cell, every hemoglobin molecule be 100% saturation all times? No. But the saturation at rest is usually pretty high, usually 97% or even above. Now, if you're exercising and working out, that saturation can drop a little bit as your body's trying to constantly breathe harder, breathe deeper to get that oxygen saturation back up. So it all depends on your physical ability and how much you're pushing yourself, how deeply you're breathing. If you're trying to gasp for breath, that's because you're trying to replenish that oxygen. So O2, or oxygen, is loaded in the lung. Sure, take a breath in, you put oxygen into your lungs. That oxygen is loaded into the hemoglobin. Once it's loaded on the hemoglobin, it's called oxyhemoglobin. That oxyhemoglobin will then leave the lungs and head out to the rest of the body, because the rest of the body needs oxygen. You'll then unload the oxygen in the tissues, whether it's muscles in your arm or maybe it's the skin of your leg or whatever. All of your tissues in your body need oxygen for the processes they run. So once you unload, it's called deoxyhemoglobin. Deoxy without oxygen. So oxyhemoglobin has oxygen. This deoxyhemoglobin doesn't have oxygen. You've unloaded. Now, do you unload every single oxygen molecule on that hemoglobin? No. You're only unloading what the tissues need, because there's a certain saturation of oxygen called a partial pressure here. There's partial pressure of oxygen in the blood, partial pressure of oxygen in the tissues. So you only unload the amount that's necessary, which means even in deoxyhemoglobin, you still have some oxygen on the hemoglobin. But you can also sometimes pick up carbon dioxide. Now, in this case, it's carbaminohemoglobin. That means you put carbon dioxide onto the hemoglobin. Now, I have down here 23% about of hemoglobin is transported this way in the blood. The vast majority is just simply in the plasma. This just helps the process along. Now, I mentioned about this oxygen saturation in your blood. What this little piece is, you can see it stuck on the finger, that's a pulse oximeter. May have had one on your finger or you've seen one used. The idea is it's measuring the amount of oxygen saturation in your blood. What's happening is it's taking red light and infrared light and shining it into your finger. The amount of red light and infrared light that then is absorbed on the other side is measured. The percentage of oxygen saturation will absorb different amounts of these lights. So the light itself is absorbed differently, having higher amounts or lower amounts of oxygen saturation. What this allows is saying, hey, we have low levels, hypoxia. So in the case of hypoxemia, we're looking at about 95% or lower saturation. Now you're saying, hold on, 95% lower, I've been at 95%. It happens, sure. That's just the very start point. Usually below 90% is when things really start getting problematic. But on average, you want to be up in that 96, 97, 98, unless your body always maintains it slightly lower. Again, these are averages. The human body is variable. You might have a unique situation where your body just runs naturally at 95%. No problems. It's been that way your whole life. That's between you and your doctor. You've seen this. You know it. No worries. These, again, are averages across the entire population. So a pulse oximeter, wavelength of light going through, measuring start to finish, how much is absorbed, it can calculate the saturation of oxygen in your blood. So a little bit about red blood cells and what they're doing in your blood. Carrying oxygen, carrying carbon dioxide. Kind of an important task.